have is a change in gene expression that can get passed down um, within the life of an individual or even across generations. And there's some evidence now that some of these epigenetic changes in gene expression can get passed down over 50 million years in some bacteria. So these can be very long-term changes in gene expression. And the way they work is that the DNA is methylated and that will silence gene expression. So say this is a piece of DNA and you have a little methyl cap that silences gene expression. And then if that methyl cap is lifted off, the gene can be expressed. Put the methyl cap back on again, and that gene is silenced. And so that's one of several mechanisms by which these epigenetic modifications can occur. And they're very interesting because they can let you look at how the environment might change gene expression and bring about a change, say, in shape or physiology or behavior of an organism, but it doesn't involve a change in gene sequence. And so this is something that can be reversed in ecological or in evolutionary time. So that's why we thought methionine would be a fun one to have a look at. So here are some broods from our polychaetes. This one is from a control, so just raised in seawater, and they have the big cannibalistic juveniles. Here's its eyes. These small dispersive larvae that can move potentially to another population. And these little yolk sacs are non-developing eggs that the big guys eat. With moms that are treated with methionine, which we think is the environmental cue that can bring about this transition from one type of larvae to the other type of larvae in developmental plasticity, if we treat moms with methionine, they make the little larvae, but if you look in that capsule, there's none of the big cannibals present. So it's only the little larvae and the nurse eggs. And these larvae will literally starve to death if you don't hatch them. The big guys are happy to eat these non-developing eggs. The little guys just swim right by. They don't recognize them as food, and they will literally starve to death. This is an egg capsule from a mom polychaete treated with folate, which is a methyl donor. And what you see is large, all the offspring develop into these very large juveniles. Again, here are the eyes right here. And so those are just some pictures of the broods. When you count them, because of course biologists like to count whenever we possibly can, here we have the percentage of larvae from a brood that are small in blue, medium size in green, or the big cannibals in this beigey color from the control broods, and then so raised in seawater, and then from moms that were cultured in very little methionine, a little bit more, so 10 to the minus 7 molar, and then more methionine, 10 to the minus 5 molar. And the controls, what you have is some little, some mid, and some large. But by the time you get to the higher concentrations of methionine, there are none of the big cannibals left, or very few of the big cannibals left. All the larvae have switched to these very small larvae. And remember, we think this is a real switch because they don't recognize the traditional foods that the cannibals would eat, and they will literally starve to death before they'll eat some of these other nurse eggs. We also found that this pattern, this transition from making equal numbers of little and big offspring to making only small dispersive offspring, was paralleled in the amount of DNA methylation in mom's genome. So this shows the global DNA methylation as a box plot. So there is like 10 females, I think, in each of these different groups. I can't quite remember, but say 10 females in each group. From seawater, low concentrations of methionine, more methionine, and then more methionine. And what you find is from control females, a low amount of global DNA methylation. But once you get to the higher concentrations of methionine, there was much more methylation within that genome. Now, this is the whole genome. We're not looking at how the presence of a methyl cap binding to a gene can influence the expression of that one gene, but it's the whole genome. And it looks very much like methionine here would be a methyl donor, but we know from work from other labs and in other compounds It's never really that simple. Sometimes there'll be an increase in methylation in one part of the genome and a decrease in other parts of the genome. So we don't really know about the specific genes that are involved, but there's an overall shift towards more methylation in this group. And this just shows a couple of these larvae that have come from the methionine treated broods. So again, the question is, the worms are living in this really bizarre habitat. They have developmental plasticity where they can make some little and some large offspring. And what is the environmental cue that can drive this transition to occur? And we think methionine is that cue. With high levels of methionine, you get lots of the small larvae. And again, they will not behave like the cannibals. It's not just like the cannibals stop growing, but they really seem to have shifted over to this small larval form that can't recognize the nurse eggs as food. But if you hatch them and you feed them phytoplankton, they grow very happily. So this is one near metamorphosis. Again, here's its lovely 3 pairs of eyes. And then they'll undergo metamorphosis and grow up and be a perfectly happy little Polydora. So this methionine really does seem to be activating a different suite of genes, driving the larvae to have a specific developmental pathway. But within that pathway, they're quite normal larvae and they will undergo a normal metamorphosis. So life in what I think is a fairly extreme environment, again, because of the continuously moving sea of mud that can erode and deposit sediments very quickly and, smother out populations or wash out other populations. A prime source of primary productivity that's very odd relative to a lot of other coastal ecosystems. And this is one example of a species that produces both small larvae that can disperse and they're like a safety net. They can go off and colonize a different part of the basin, or large cannibalistic juveniles that can recruit right next to mom and dad if it's a suitable habitat for them to do that. And I'm just talking about one species, but there's actually 3 species of polychaetes that I know of in the Minas Basin that do something similar, and 2 species of Ascoglossan episthabranchs. This form of developmental plasticity is the scientific term is called poisologeny. It's extremely rare. There's like 30 species, 28 or 30 species worldwide that do that. And so for this small number, like 3 polychaetes and 2 Ascoglossan episthabranchs to have a variation on this theme all in one geographic spot just shows that it's an indication that it really is a very dynamic and extreme environment. And so we're working on the hypothesis that methionine in the marsh grass is a cue to mom that can change her epigenetic profile, but then also bring about a transition in the way her larvae develop. Okay, so just quickly, in the last few minutes that I have, I want to talk about another area of research that we've been engaged in, and that is looking at the effects of microplastics on marine invertebrates. And we hear about microplastics all the time in the news as well. Fun fact, not really, we all need tissues in a minute. 350 million tons of plastic produced annually. It's a petroleum product. Very little of it goes into the recycling stream. And of the stuff that goes into the recycling stream, a tiny, 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 tiny bit is actually recycled. It's really very sad. And so, Sally, can you put up our next poll, please? Which type of plastic is the most persistent in marine ecosystems? Just before we talk about plastics, let's just think about how long they last. So here's your question. Which type of plastic debris is the most persistent in marine ecosystems? Plastic bags, 6-ring plastic beverage holders, disposable diapers, fishing line and plastic bags. Nobody's biting on the 6-ring plastic beverage holders. Fishing line, plastic bags. Oh, you guys are doing so well. We'll just give you another minute to keep voting. 85% have voted. Anybody else going to vote? Oh, another vote for fishing line. What about plastic bags? Anybody going to vote for plastic bags? Okay, so fishing line won the poll. Um, Plastic bags a little bit further behind, and not many people voted for the disposable diapers or the 6-ring plastic beverage holders. Whew. Okay, so here are the data. Oops, let me just make that go away again. And so this is from one of my favorite sites. It's called Our World in Data. And if it's not a site you know, you should go look it up because it's super fun. Fishing line, decomposition rates of marine debris items, fishing line, polypropylene, 600 years, plastic bottles, which would be polyethylene and polycarbonate, 450 years, disposable diapers. I don't know what they're made of, but look at that, 450 years for disposable diapers. Who knew? The plastic beverage holders, 400 years, and plastic bags are way down here at 20 years. And so there's some of this plastic is extremely persistent. And a lot of the biodegradable plastics we buy now are actually plant-based, but they still have petroleum polymers that hold them together. So sometimes when they break down, they just break down into microplastics that we don't see, but they're very persistent. So just because something tells you it's biodegradable, it may be, it may not be. And here's a fun little piece of information. This is from the Marine Pollution Bulletin, where they did a survey of microplastics in subsurface seawater off Vancouver Island. So Vancouver Island is here. And they used plankton nets that could capture particles larger than 60 microns. So they didn't get the nanoplastics, but they could certainly get a range of the microplastics. A lot of these samples had low amounts of microplastics, but by the time you get into the strait between Vancouver Island and the mainland, you're getting up to 4,000 microplastic particles per cubic meter of seawater. And up here by Haida Gwaii, where you'd think the water would be very pristine, we're up to 9,000 particles of plastic per cubic meter. This is an extreme study. We don't usually see this in coastal waters, but there's a couple of cautionary tales. We miss the nanoplastics when we do these sorts of surveys, and areas that can look very pristine sometimes are not. So we followed up on looking at microplastics in the water column, and we used studies, sort of amount of plastic that was down here and an amount of plastic that was down here. So fairly low on that particular scale. But before we talk about microplastics, let's talk a little bit about plastic leachates like bisphenol A and its safe alternative, BPS, bisphenol S. So a lot of the BPA-free plastics we buy right now have bisphenol S or bisphenol F in them as a replacement, and they're almost chemically identical. And how much bisphenol A is found in aquatic systems? A lot. And so this is from a review article from a few years ago, where they surveyed results from the literature from river water in Japan and the Netherlands down to drinking water in Germany, and found this huge range from 100 micrograms per liter down to a very, very low amount of BPA. And so we didn't know where to start with our study, so we just went the whole range from 300 micrograms per liter to a tiny, 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 tiny bit of a microgram per liter. And I'm just going to jump to this slide. These images here show some of the polychaetes that Melissa was working on undergoing regeneration. This one is the control. So here's the original segment and the regenerating tissue, nice little thorax, nice little head. This one is at the same age of regeneration, but was exposed to BPA. And this one is at the same age of regeneration, but was exposed to the safe alternative, bisphenol S. And you can see that both are very, very stunted. And so if you compare the lengths of the regenerates, the controls have nicely sized blastemas, but those exposed to BPA and BPS all have regenerating tissue that's much, much smaller. And just look at these concentrations. So this is 0.3 micrograms per liter down to 0.0003 micrograms per liter. So we're exposing our polychaetes, to concentrations of BPA that are found in rivers and coastal areas in different parts of the world.